Thank you very much.
Thank you very much, Andrea, and thank you very much, and good morning, ladies and gentlemen. I would like to take this opportunity, Gavin and Ravi, and the organizing committee for me to come and visit all of you. I was asked this morning to talk about the anti-fibrinolytic, particularly the Aprotinin story. First, I need to declare I have no conflict of interest of any industry for the last 15 years because I sit on a good number of government committee in Ontario. I came from London. I always have to explain London, Canada, instead of London, England. I moved from Toronto to London about 10 years ago. We have about the complex London Health Science in St. Joe Healthcare, the 3 hospital, 2 health science complex, an operating budget about $1.5 billion per annum, 42 operating room, 1,400 cardiac surgery, and a lot of robotic and minimally invasive surgery, one of the major centers in Canada. My department now have 80 consultants, 12 fellows, and 47 residents. So the bar trial or the anti-fibrinolytic, I'm going to very briefly describe to you about the evidence-based efficacy and the safety, and to highlight along the pathway and the journey, bring you back to the early days of Aprotinin until the current status. This hit the newspaper and the media. This is from the ABC News. This is also from the New York Times, and also back in February 14, 2008. This is hit the news media that patient had died unnecessary because of Aprotinin in the U.S. market. That's because of the medical legal during that time in the 2008, many class action are being grooming in the background with a high evidence and consistent evidence that there are morbidity and potentially mortality, but more important is the kidney failure are consistently being found on Aprotinin. Bring you back to some of those mechanisms. So this is one of the drugs that we use routinely in cardiac surgical procedures. The Aprotinin in red and the black here, and the gray is the lysine analog, which is the TA or the Amica. You can see here the Aprotinin actually have a vast mechanism of action. The original medication being tested for Aprotinin is not so much for bleeding, it's actually for lung water, for anti-inflammatory effect because of its mass effect on the coagulation and the anti-inflammatory pathway. The lysine analog for the TA is more restricted on the plasmin and the plasminogen for anti-fibrinolytic aspect. So David Royston is fundamental in the seminal paper on the Aprotinin, the use of that in cardiac surgical procedure, because it was found that by accident or by observation that actually is reducing blood transfusion and blood loss in cardiac surgical patient when they're measuring lung water in anti-inflammatory. And it never, and since then, the company invests, and in 1993, their proposal to the FDA, you look at all those are TA versus control group only, placebo. These are randomized double-blind controlled single-center trial, multi-center trial, and they submit those for FDA approval back in 1993. And those are only when the first use of a patient for cardiac surgical procedures, high risk for bleeding, not simple procedures, but high risk for bleeding. And you can see demonstrate a reduction of the blood transfusion rate, the incidence of exposure, from about 77% to about 50%, 42%. Bearing in mind this is the early 90s, the placebo transfusion rate about 70% at that time during the submission. And those are for redo surgery, combined procedures, and combined repeat procedures. So those are in the patient with high bleeding risk for those. Even back in the 1993, we don't see that as much. The data actually was buried in the FDA submission. Even when the companies submit the data, there are an observation, a 3% increase of kidney failure for the Aprotinin, while the kidney failure in the placebo group, despite more bleeding and transfusion, is only 1%. There is already from 3% to 1% kidney failure rate. Also, there is a creatinine or renal insufficiency rate from 12% to 23% buried in the submission of the FDA back in the 1993. Nonetheless, the aprotinin was approved by FDA back in December 1993, used for high-risk patients, repeat cardiac surgical procedures in primary CABG operation. Subsequently, more and more data coming out. Aprotinin has been used in other settings too. The exposure also regarding the hypersensitivity or the some of the anaphylactic reaction. You can see here they are less than 6 month exposure. They are a high risk for the anaphylactic IgG antibody exposure. So there are reactions. So when you're given aprotinin to the patient, you want to bring them back for take back. You might want to be careful not to give another big dose of aprotinin because of the hypersensitivity reaction. So the company is doing very well with that medication, moving along with the high risk, but I guess maybe in the medication they want to expand their use. So they put in a new submission back in the 1996, 3 years afterward, to look at not just for high risk, they want everything. They want even the low risk patient. So they're looking at the primary bypass operation compared to placebo. These are aprotinin compared to no medication for the cardiac surgical procedures. And obviously, they demonstrate a reduction in the blood transfusion. So the FDA indeed approved. This is efficacy study. This is not safety study. So the approval for the drugs is for the reduction for blood transfusion or exposure to the allogeneic blood transfusion. So it was in August 1998 was approved, expanded the indication to routine primary bypass operation with a black box warning for anaphylaxis. Many papers came out from that time for the there's a lot of forest plot looking at the chest re-exploration, high morbidity, even associated with mortality, consistently telling you that aprotinin is beneficial in reducing the incidence of chest exploration. Mortality for aprotinin versus placebo, no medication, are basically not much different in regarding the mortality rate for all the study as well. So why the aprotinin back in the 2006? So they have a good 10 years or more of the use of aprotinin in cardiac surgical procedures. Not until Dennis Mangano on MedSpy, which came from the 52 center around the world. I'm actually at that time organizing for the Canadian MedSpy group. And it's a very enriched database from all countries looking at all this data. So Dennis Manganos and the MedSpy look at all the data for blood transfusion and look at aprotinin and anti-fibrinolytic and make that statement and publish in a very high impact journal that really take the media and the scientific world by storm. The conclusion is continued use of the aprotinin is not prudent. Why he say that? In this large observational study, despite it's a unadjusted analysis but being criticized, there are control TA, AMICA, aprotinin group, and these are all the group of patients, 1,200, 800 patients, and he consistently showed a dose response for aprotinin causes mortality, associated with mortality, associated renal failure, cardiovascular event, and the composite event. In addition, not just one paper, in the same year, Kevin Kakuti from Toronto General, which we have the database that I set up for a good number of years now, also look at a propensity score matching as much as we can, going through the univariate and multivariate to look very specific of the factor that you can appropriately match. Out of those papers too, looking at the aprotinin versus TA, very well matched 449 patients with all the baseline characteristic. You can find here there's no difference even for transfusion rate. So when you match the right characteristic for high-risk cardiac surgery procedures, TA and aprotinin, there's no difference for efficacy, similar efficacy. What the difference is, there is still the renal dysfunction with aprotinin. So with the Mangano paper in New England Journal, and the transfusion paper from the Toronto General that we have at that time totally turned the literature upside down, and FDA right away called for the first meeting to look at the safety data. That is back in September 1st, 2006. So 2 papers came out in May, and in July, September, FDA called for a meeting. A safety meeting by the safety panel. The committee they call are the Cardiovascular and Renal Drug Advisory Committee. Only one committee the first time to discuss the safety in September 21st. There's no decision at that time, and very quickly, within a week, September 27, a week after the Bayer company went to the FDA, they suddenly, in about a week time, go back to FDA in private and say, actually, we do have our own data. It's the I3 data that we have studied over 60,000 patients. And actually, we haven't shown you those data. You can imagine how mad or how displeased FDA is. So in September 27, is right away, FDA called for another emergency meeting for the subsequent year and want to thoroughly investigate and look at the data for Dennis Mangano, Kevin Kakuti, and the Bayer Company I3 paper. And right away in the December, they slapped on a restriction to Aprotinin 2006, i.e. only limitation in focus is now indicated for only patient for high risk. So they reversed the 1998 for all patients, bring it back down to only high risk patient. In addition, a new warning was put in, renal dysfunction for Aprotinin, and also showing the anaphylactic reaction. So December 2006, right away they put it in. These are the data that Bayer didn't show the FDA in the first meeting. These are their own data, 66,000 patients. Conveniently forgot to mention that to the FDA. And this data look at the outcome for the risk, relative risk, 95% confidence intervals, mortality, 68% increase, all across the midline, 1.53 to 1.8. Acute renal failure, acute heart failure, stroke, they're all positive in their own company database. When the FDA is assembling the committee to review all this data, more and more people came out. Dennis Mangano looked at the 5-year follow-up and published in JAMA of the original paper, also indicating Aprotinin in the 5-year survival the mortality are continuously much higher than the 2 lysine analog. Another paper from Surgical Publication 2007, a few months after that, major center cohort study, also looking at 3,000 patients, also demonstrate renal dysfunction. They keep telling us the same story, reaffirming both the observational study that there is problem with Aprotinin. And then Brown also looked at 138 trial on the meta-analysis and also demonstrate there's no difference in the efficacy, but there is a consistently higher dose with the renal dysfunction. So every single different center, different population of the surgical patient telling you the same thing. So September 12th, 1 year after that embarrassing episode when Bayer didn't show the data, this time FDA called in 2 committees. The committee actually is the Drug Safety and Risk Management Advisory Committee. This committee have the authority to pull the drug out of the market. So they are not just asking for one committee to review this, they have 2 committees sitting together. This is the original committee, the Cardiovascular Renal Drug Advisory, and the Drug Safety. This is to look at the approval of the Aprotinin, focus on the investigation and the analysis of the 3 trials. And this time the FDA do their analysis. They took all the raw data. They asked, that's why it took a year, because they asked for company and the 2 center to send them all the raw data to analyze. So when they look at all the data, I bear you the detail. This is what the FDA reanalyzing all the data, different from all the 3 company, or the company and the study. Mangano paper, relative risk on control. Aprotinin versus control, You can see here renal failure crossed over the 1. Renal failure is there again. All the other one, renal composite again, there's a signal that renal failure is a problem for aprotinin. All the other one for the cardiovascular morbidity, there's not much p-value or significant. Kakuti paper on TA, relative risk, renal again, renal failure. Cross the midline, so we can't talk about the other one, but renal function is always a problem. The company own data are looking at aprotinin versus TA or AMICA. Every single one of their outcome in their own company database have higher risk, morbidity and mortality, which they are not sharing with anybody. During that meeting in September 12, Kevin was also asked to discuss some of this data, and the FDA have asked him beforehand to look at his own data and also revisit 3 models, to look at the model, because sometimes when you do a propensity score, if you're missing some of the non-independent factor, like say transfusion, if it's associated with morbidity outcome, you need to make sure that you control that part too. So there's 3 models being put in, another mathematical model that put in to adjust for those. Again, mortality, model 1, model 2, model 3, MI, stroke, renal failure, renal disease. Consistently again, during the FDA presentation, aprotinin causes renal dysfunction and renal failure. So the vote at the FDA during the 2 meetings, because at that time, the BAR trial is not published yet. The largest randomized control trial is being wrapping up. So the FDA asked for a vote. Tresilol or aprotinin risk and benefit in their control study, and your consideration with only now the observational study, do you recommend continuing marketing? As I mentioned, they want the safety committee there. The question posed to them, do you recommend marketing authorization? 16 yes for them to continue. 1 say no, 1 abstain. That's because they're pending and waiting for the BAR trial. Nobody want to make a move on the observational trial. They asked the second question. The I3 drug safety and all those suggest disadvantage. Should this finding be described in the product label, i.e., should they force the company, even moving forward from now as of September 12, 2007, should we need to put more label? There are some controversy there. You can see more no here, voting no, and yes. So, but it's 6-1, 6-11. When they asked, should the Bayer company or the literature have more randomized control trial? This one is very easy. It's a no-brainer. Everybody say yes. So the question though is observational study. All those trials, the 3 major trials lead to the demise of aprotinin are all observational study. So best evidence for efficacy always is RCT. It's the gold standard. However, the harm is always by large observational trial. RCT is not ideal for looking at those unless you have a major cardiology trial or the BAR trial to address. So mountain of evidence out there, the bridge to practice. How is aprotinin in compare to TA or AMICA? Right after the meeting, September 17, let me remind you, the FDA have that decision. October 16, the safety board of the BAR trial sent to all the investigators indicating we are writing to recommend because the all-cause mortality are increasing in both the aprotinin versus the other 2 medication. So all the BAR investigators, we talk about stopping the trial, and we did. The trial was stopped, and we indicating the trial even moving forward for the complete analysis. There is an absolute increase in mortality of 2%, i.e. the number of harm is 50 patients. So every 1,000 patients will be continue doing the trial in high-risk patient, high-risk bleeding patient. Aprotinin given will have additional 20 preventable deaths. There is an alternative. This trial is not talk about placebo. The trial is talk about an alternative that have also reduced blood transfusion, similar efficacy. So the investigator will think that it's not prudent for us to continue. All of this despite a reduction in bleeding. But the efficacy for blood transfusion is no different. So with that, we inform the FDA when the trial being stopped. So in October 19, FDA was informed. October 16, the letter was sent out. 3 days after, FDA act. FDA right away called this investigation, label it, all the things that the bar trial shown to them. 30-day mortality is nearly reaching the conventional statistic. There is a number need to harm, 2% absolute mortality rate increase, and there are other complications. So with that, FDA slapped on another label. And also, November 5th, a month and a half after, request the market suspension of Tresilor in Health Canada and Germany as well. I think Germany take action first, and then we all add. One of the official on the FDA indicate, FDA could not identify a specific patient population at that time, in the time of 2007, that any benefit of using aprotinin outweigh the risk when there are good standard of alternative for efficacy. Right away after 2007.
I'm afraid some other group, I think, need the room. So
Okay.
I'm sorry to rush you, but I think we
I'll give you the bar trial. So more paper coming out. The bar trial is the high-risk patient, 18 centers, close to 3,000 patients. The dosage we talk about, double-blind randomized, actually triple-blind randomized. The dosage.
I'm very sorry to interrupt. Gavin, can I just suggest that maybe rather than rushing this section just now, we put it at the front end from 2 o'clock for a 10-minute session to allow the lunch boxes to move around?
I have another session. I have to present in a minimally mitral.
We're working your hard, Dave. That's the way it has to be. But we're going to have to let the lunch boxes go around, otherwise we've got people waiting.
Okay.
David, can you wrap up in a couple of minutes?
Yeah, I'll give you a conclusion. So the primary secondary outcome, very well matched because it's a randomized controlled trial. So that you don't have to do any risk adjustment at all. The key aspect is the patient surgical population, very well matched. Bypass other procedures, very well matched. Primary outcome, efficacy, no different, cross the midline. Aprotinin versus other gold standard, no different. Survival is aprotinin is worse compared to the other one. The cause of death, the key aspect, this is all adjudicated cause of death, primary cause of death. Aprotinin still are significant compared to the other one. For any cardiac cause of death, aprotinin still significantly higher than the other two. When you look at the CRU data, CRU and adjust, aprotinin 30-day mortality are harmful compared to the lysine analog. All the other efficacy outcome and other complication, there's no different. Interpretation, 2% increase, 50%, 50 patient, 1 additional patient. Despite aprotinin indeed reduce your bleeding, there's no doubt. So what bar doesn't tell us though, that bar doesn't tell us about pediatric patient, non-cardiac patient. So we cannot use bar to apply to other population. But what do we learn from bar? Bleeding and anti-fibrinolytic are very, very emotional issue. Important for large trial with meaningful comparison and outcome. Efficacy is easier to prove than safety. Observational study may not provide comparative benefit. Meta-analysis may be sometimes overemphasized. So what is the editorial accompany that publication? That we need to make sure that we have the right outcome and safety outcome in any major trial. Efficacy as of 2008, aprotinin efficacy the same, but there's harm effect. FDA in May 14 basically remove all the stock. 2001, the most recent one, David Henry, on mortality, again, these are all RCT, randomized control trial. The I-square is 0%, which means every single randomized trial telling you the same story. Aprotinin still increased the mortality compared to all the other ones. This is 2001 last year, major meta-analysis. Kakuti, we just published another one on the risk, try to do another propensity score. Again, acute renal failure are higher in aprotinin despite the matching. When he doing further subgroup analysis. We talk about two subgroup now, two stages of statistic. On the very high risk group, maybe some signal for beneficial in aprotinin. So in concluding the aprotinin study, the anti-fibrinolytic drugs are effective, all are, in reducing bleeding. An observational study, bar chart, is the current largest RCT ever existing in the literature, showing an increased mortality rate despite benefit for blood loss. And anti-fibrinolytic agent we talked about this morning is not just a substitute for blood transfusion, the whole systems. So what do we learn, as this was posted up in 2001, that in the 1A lysine analog, we need to use that. It's a 1A evidence and recommendation. Class 3 recommendation is a negative recommendation for using aprotinin, still in 2001 aspect. Health Canada and Germany just released the marketing, not to encourage the use, but allow it, but with a very strict definition, only for high risk. So they basically swing the whole thing back to 1993 recommendation, only for high risk patient. FDA still have not allowed it to happen. It's still not available. This is as of yesterday, a download from FDA. It's still not available. So, since I'm in England, these are the opinion upon my facts. Thank you very, very much.